Imagine this, one tiny typo, just one single letter flipped in your 3 billion letter DNA instruction book, quietly sets the stage for cancer 20 or 30 years later, or makes high blood pressure sneak up on you, or even explains why losing weight feels impossible no matter how hard you try. Terrifying? A little. But revolutionary? Absolutely. Google DeepMind just unveiled AlphaGenome, an AI that reads up to 1 million of those DNA letters at once and predicts exactly how those typos mess with your body's controls. And here's the wild part. Since its preview launch last June, nearly 3,000 scientists from 160 countries have already jumped in, hammering the system with about 1 million API calls every single day. The full peer-reviewed breakthrough landed in Nature on January 28, 2026, literally days ago. By the time we're done here, you'll see why experts are calling this the AlphaFold moment for the dark genome, and what it could mean for your future health. Buckle up, this is next-level stuff. Welcome to AI Observer. Let's decode human life with AI. First, a super quick DNA refresher so we're all on the same page. Your genome is the master blueprint. 3 billion chemical letters A, C, G, T twisted into the famous double helix. But only about 2% of it actually spells out the recipes for proteins, the actual building blocks like muscle fibers, enzymes that speed up reactions, hormones that regulate your mood and metabolism. The remaining 98%, long dismissed as junk DNA, now we know it's the dark genome. This is the command center. Switches that flip genes on or off like light bulbs, timers that dictate precise activation moments, volume knobs that ramp expression high or low, even 3D folding maps so distant DNA segments can loop together and collaborate. Picture the protein coding parts as the raw ingredients in a massive family cookbook passed down generations. The dark genome is every scribbled chef's note. Simmer exactly 12 minutes on medium heat. Add chili only for spice lovers, skip for kids. Bake at 350 degrees, no higher, or it burns. One smudged or torn note and the dish could overcook into a charred mess. Think uncontrolled cell growth, turn out underdone and flavorless, or never make it to the table at all. Here's why this dark genome is such a big deal. Over 98% of the genetic variations tied to everyday diseases, various cancers, type 2 diabetes, Alzheimer's, obesity, lurk right here. Not in the protein recipes themselves. These mutations don't shatter the ingredient outright. They corrupt the usage instructions. One variant might force a growth gene to blare non-stop, leading to tumors. Another could silence an insulin handler, spiking diabetes risk, or subtly alter brain neuron signaling. For decades, decoding this was a nightmare. Scientists could only scrutinize tiny DNA snippets. It felt like piecing together an epic blockbuster film from 5 scattered 10-second clips, no context. AlphaGenome flips the script entirely. This model is engineered from the ground up for scale. It ingests up to 1 million DNA letters, a vast, unbroken stretch, and delivers thousands of functional predictions with pinpoint resolution. That covers everything. Gene expression strength, RNA splicing edits, chromatin accessibility, histone modifications, transcription factor binding, even 3D contact maps, all computed simultaneously. Trained on enormous human and mouse datasets, it outperforms top prior models in 25 out of 26 benchmarks. Gene expression forecasting, boosted by 14.7%. Speed Demon, it assesses a mutation's ripple effects in about 1 second. 1 second versus days or weeks of painstaking wet lab experiments. And get this, it runs efficiently on just a single H100 GPU, making it accessible for labs without supercomputers. Under the hood, AlphaGenome's architecture shines. It starts with convolutional blocks that scan the DNA like a high-res zoom lens. Then transformer blocks process the big picture, using attention mechanisms to connect distant parts of the DNA, much like your brain links ideas. Let's lock in the concept. Your genome is the largest library on the planet. The dark genome is the librarians, lighting crews, and security teams. A mutation? It's a rogue librarian hauling an explosive thriller into the kid's corner at midnight. AlphaGenome steps in as the ultimate AI sleuth, mapping the entire multi-story complex in seconds, spotting the flickering bulbs. It infers unspoken rules, connecting dots no human could trace, almost like the AI is learning the grammar of DNA. Now, dive into a hands-on demo walkthrough. Pretend you're a cancer biologist tackling T-cell acute lymphoblastic leukemia, TL, an aggressive blood cancer that hits kids hard. You suspect non-coding mutations near the TAL1 oncogene are the culprits, hijacking normal activation. You load AlphaGenome with a 1 million base segment containing one such variant. The AI crunches normal sequence against mutated. In under a second, it outputs. This precise change forges a fresh binding pocket for the MYB transcription factor, which then supercharges TAL1 expression by 7.5 times specifically in early blood progenitor cells. It recreates the exact disease pathway. No prior leukemia training needed. The model just mastered the dark genome's syntax so thoroughly it extrapolates the mechanism from basics. This isn't mere guessing, it's deep insight. Now scale that. Screen thousands of patient variants, score drivers versus harmless passengers, fast-track wet lab validation and targeted therapies. Extend that to more examples. Splicing glitches. In spinal muscular atrophy, SMA, a neuromuscular disorder, or some cystic fibrosis forms, faulty RNA splicing mangles protein production. AlphaGenome forecasts splicing shifts across tissues with sharp precision. Input a patient's variant. It highlights the disrupted splice junction, paving the way for custom antisense oligonucleotides, short RNAs, to patch the edit, or genome-wide association studies. GWS. These mega-analyses flag DNA spots linked to traits like adult height, obesity propensity, hypertension susceptibility. AlphaGenome refines 49% more credible hits by predicting effect direction, upregulates or downregulates, transforming vague correlations into concrete biology, unearthing novel druggable pathways. It's also shining in neurodegenerative research, like pinpointing variants that dysregulate brain genes in Alzheimer's, or infectious diseases, where it models how viruses integrate into host DNA controls. For tissue development, it maps essential non-coding strands for nerve or liver cells, revealing why mutations derail organ formation. And in synthetic biology, it designs custom DNA with tailored regulatory functions for new therapies. DeepMind's legacy amps this up. Start with AlphaFold, their 2020 bombshell for protein structure prediction. Proteins aren't flat recipes. They fold into intricate 3D shapes, loops, sheets, helices that dictate function. For 50 years, predicting those from sequence alone was biology's holy grail. AlphaFold nailed it using deep learning on known structures, scoring 92.4 median GDT in Casp14, the Protein Olympics, basically human expert level. It predicted shapes for nearly 200 million proteins across all life forms, open sourcing the database. Impact? Accelerated drug design. Companies like Pfizer used it to model COVID variants. Biotech firms targeted Alzheimer's plaques. By 2024, over 1.5 million research citations, earning Demis Hassabis and John Jumper the Nobel in chemistry. AlphaMissions followed in 2023, classifying 71 million possible protein-altering mutations as benign or pathogenic. Now AlphaGenome builds on that foundation, shifting from protein hardware to regulatory software. It's especially game-changing for rare Mendelian disorders, single-gene devastators like Huntington's or certain epilepsies, where big-impact variants hide in non-coding shadows. Identify culprits quicker, craft intervention sooner. Quick pause. Knowing AlphaFold cracked proteins and AlphaGenome now unlocks controls. Does that make you more optimistic about curing genetic diseases? Comment game changer or still skeptical below. Ethics check. Power like this requires vigilance. AlphaGenome forecasts molecular tweaks, e.g., mutation ramps expression in kidney cells. Not ironclad fate, heart attack at 50. Environment, diet, exercise, other genetics, randomness, they rule. Risks: laypeople misreading output spark anxiety. Data biases toward European ancestries could shortchange global accuracy, exacerbating inequities. Genome privacy breaches, catastrophic for insurance, employment. Think real cases where genetic discrimination led to job loss, despite laws like GINA in the US. Direct-to-consumer tests like 23andMe already raise hacks data sale fears, slippery slopes to embryo editing or eugenics if mishandled. DeepMind wisely made it non-commercial research only, with free API and GitHub open source, empowering community oversight, bias corrections. But urgent, enforce robust international guidelines echoing post-AlphaFold calls to prevent global divides where only wealthy nations benefit. Future's buzzing, with 1 million plus daily API hits and rapid uptake. Envision CRISPR AlphaGenome hybrids editing dark genome flaws pre-symptom, synthetic biology crafting bespoke DNA regulators for therapies, even simulating disease routes to test virtual cures. Experts hail it as an incredible feat and major milestone in genomics. As DeepMind's Natasha Latasheva notes, it's bridging toward a unified code of life. Proteins, regulations, all harmonized. Poll time. Thumbs up if AlphaGenome plus AlphaFold excites you about beating diseases. Thumbs down if ethics privacy weighs heavier. Share your top takeaway. Would you genome sequence with these tools? I reply often. If this enlightened you, like, subscribe for AI biotech deep dives, bell on. Links, Nature paper, GitHub, DeepMind API, description. Thanks. Medicine's future race is ahead. We're tracking it. See you next.